So a virus walks into a bar, and the bartender says, "We don't serve viruses here." The virus replaces the bartender and says, "Now we do." All right. Sorry about that. Okay, so let's get on to tonight's discussion. Do not take this as an insult, but all of you are a 100 trillion cell or sack of cells. You have 20 or so bacterial cells for every one of your mammalian cells. And so the question really does arise of who is the host of whom. Several pounds of what you think you are comprised of microbial cells. They're in your gut, they're in your mouth, they are on your genitals, they are in your skin, they are in your airway. Most of the time they get along with each other. And most of the time they get along with you, but not always. And more biomedical researchers are discovering that the microbiome, those hordes of lifelong inhabitants in and on us, are central to health, disease, really just about everything that's important to us as living creatures. And, for example, how we respond to antibiotics and other medical treatments. There are amazing harmonies amongst this overall bacterial world in you, the human microbiome, but those harmonies can be thrown out of whack, and we will learn a little bit about that today. We have with us today, I'm happy to say, two discussion leaders who will help us understand better the ways of our bacterial stowaways. First up will be Liliana Losada of the J. Craig Venter Institute in Rockville, which has been on the forefront of identifying and characterizing the vast microbial diversity throughout the environment. She will describe the big picture, the microbiome, this diversity and plethora of bacteria in and on us. And following her will be Alison O'Brien, former president of the American Society for Microbiology and professor of microbiology at the Uniformed Services University of Health Sciences. That's in Bethesda. The J. Craig Venter Institute is in Rockville. So this is local talent. And Alison will drill into one of the many species, Escherichia coli, she might pronounce that differently when she comes up, which live in us, but in some versions and under some conditions can wreak havoc. And once Liliana and Alison have finished with their opening remarks, I will facilitate the discussion, and hopefully we'll have a better behaved microphone at that time, but I can't guarantee it. And so with that, let me ask Liliana to come up and tell us what's on her mind.
Thank you, and thank you for inviting me. Thank you, everybody, for coming. I thought I would start by giving you a sort of a brief overview of how we started studying the microbiome and the technologies that we use for studying the microbiome, and then move into just giving you some of the very broad conclusions that we've so far arrived at from those studies. So to me, really, the study of the human microbiome started when Louis Pasteur postulated his germ theory of disease that states basically that disease was caused by microbial agents. And so since his first postulation of that hypothesis in the 1850s, 1860s, several decades after that, many scientists, many microbiologists started to actually investigate the microbial basis of disease and found out that, lo and behold, many of the human diseases were in fact caused by microorganisms, and that not only were those microorganisms potentially causative agents of disease, but they actually also live in the human body. And certainly at that time they weren't able to understand to what extent our human body is really a supra-organism composed of maybe 10 to 20 times as many microorganism cells as human cells. So through the course of those investigations, as I mentioned, microorganisms were found in all of the different parts of the body. At that point, several of the locations of your body were considered to be sterile, but that concept has since been questioned because we have now technologies that allow us to actually probe and study those locations and actually question whether they are sterile or not. So from the studies of the usual technology that was used, the techniques that were used to study the disease-causing microorganism would have entailed a laboratory full of microbiological media, somebody actually taking a sample from a human being and swabbing it or placing it into liquid broth and seeing whether any microorganisms grew. What became apparent after several decades of research was that there were some microorganisms that we couldn't grow in the laboratory and were in fact unculturable, and they are still unculturable. We just cannot simply put them in a petri dish, as you were saying, and have them grow. They just don't grow. And so from that, to me, came the concept of trying to really understand and trying to devise a methodology that will really allow us to understand the full diversity of microorganisms in a human body. At the same time as the notion of unculturability was arising, also parallel to that was the development of DNA sequencing technology. And DNA sequencing technology then granted the possibility of looking at the microbial diversity without actually having to culture the microorganisms, because you can look at the DNA. We know that the DNA encodes for the genes that makes those microorganisms, that allows them to do their physiological and metabolic capacities. And so if you were able to understand the DNA and the genes that the DNA encodes, then you could understand what sort of capacities are in any particular sample without having to culture the organism. So as a consequence of both of those concepts coming together in the late 90s and early 2000s came the possibility of sampling ocean water in the case of JCVI, or human sites in the case of the Human Microbiome Project, to actually try to use sequencing technology to understand all of the diversity and put them together. So the first study of a whole microbiome using DNA sequencing approaches was done on the Sargasso Sea and was published by scientists at Tiger, now JCVI, and showed that there was the technology to do such a study, to actually analyze the microbial diversity in samples. So at the same time as all this was happening, the human genome had just finished being sequenced. And now the obvious question is, well, we know there's hundreds of millions of bacteria in our body, microbes. We don't know anything really about them. Now we have to sequence all of those organisms as well. And so then was the birth of the Human Microbiome Project in the United States, and also across multiple consortia of countries around the world that have also initiated their own microbiome projects. So the Human Microbiome Project in the United States is funded by the U.S. National Institutes of Health, and has devoted to 4 different body sites, one studying the oral nasopharyngeal cavity, so basically your airway epithelium, looking at your gastrointestinal tract, genitourinary tract, and then your skin microbiome. Now, what have we learned? We've learned, as Ivan was alluding to, that quantity-wise, there's 100 trillion microbes in your body right now. We've learned that there's about 100 times as many microbial genes as there are human genes in your body right now, which means that the richness of the genetic capability in your body is not just your genes. And what would argue that what makes you isn't necessarily only your DNA, but also all of your microbial DNA that you carry around with you. So that's one thing that we've learned. Quantitatively, we've learned that. We've also learned that the microbiomes in each one of those locations are different. So what you have, the residents in your skin microbiome are actually different from those that live in your nose and your mouth. Those that live in your mouth are different from your gut, so forth and so on. We've learned that the microbiomes change from one location to the other. One of my favorite papers about the microbiome was studying the elbow microbiome. Some of you might have read it. The fabulous conclusions from that was that the microbiome from the inside of your elbow is very different from the microbiome of your outside of your elbow. They're only 2 inches away, but they're very different from one another. Right? And if you think about it, they're actually very different environments, right? One is very moist, you keep it closed, it's not exposed to, constant chafing by clothes, etc. The other one is very arid, very callous side of skin. So it is natural that they would be different, but we just didn't know that to begin with. We know that the microbiomes in your upper gut are different from the microbiomes in your lower gut. They have different adaptations, they have different physiological roles, and they help your body in different ways. And so not only do we know that they change body site, they can change over time, so that the microbiome of a woman's skin actually changes over the course of her menstrual cycle according to the levels of estrogen and progesterone in her skin. So differences in environment can also affect the microbiome that is in any one particular person. We also know that the microbiomes are different between one human being and another human being. So I'm trying to just portray to you now the complexity in the microbiomes is not simply like, oh, here's Staph aureus or here's Staph epidermidis. No, there's a whole slew of organisms that live in you, and they can change over time, and they can change from one person to the other. Now, all that being said, it would be absolutely impossible to study anything if it was every single person totally different. We've also come to understand that there does exist a core microbiome for each of these sites that is shared between people and, of course, in yourself, right? So these organisms, many of us will have, almost all of us will have Staphylococcus epidermidis on our skin, for example. Now, the quantity of that relative to other microorganisms might differ in different people. And so the structure of the microbiome is what changes mostly between people. The other thing that changes among people and between body sites are the microorganisms that are less common, the ones that are rare variants, as we call them. So they're just not very common in many humans, but they can transiently colonize you, and some of them are there permanently, but they're only in you and not in your brother or your sister or your kids or your husband or things like that. We've also then learned that in addition to a core set of microorganisms, there is also a core set of functions that those microorganisms can conduct. So in your gut, for example, in your large intestine, the microorganisms are involved in synthesis of vitamins and other essential nutrients that are necessary for your body function. And that synthesis doesn't have to happen by one single species or one single type of organism. Actually, it can be any one of a series of types of organisms. As long as one of those is present, then your body's functioning just fine. I had this list of things that we had learned, and now I forgot. So variability, a core. We've also learned, okay, so one of the major goals of the human microbiome projects has been to actually try to understand whether a microbiome has a role in health promotion, disease prevention, or disease progression. And what we've seen in the cases of disease microbiomes, we've seen many studies of disease microbiomes, including obesity, liver disease, alcoholic liver disease, type 1 and type 2 diabetes, cystic fibrosis. I mean, we can go on and on and on. But what we observe in the cases, any one of those cases where we've studied disease microbiomes and compared them to normal microbiomes, is there is a very significant difference that can be predictive of the disease condition. And we also observe that in most of those cases, the diversity of those microbiomes has collapsed, so that the microbiome, where it's normally in the normal individual very diverse, you have a nice transient population that comes in and out, and you have a robust flexibility in that microbiome. In the disease cases, it ends up being much more static and much less diverse. And studies coming out of autoimmune conditions such as type 1 diabetes, or in the cases of inflammatory bowel diseases, and also in cystic fibrosis, what we've seen is that that collapse and that static microbiome actually leads to an increased inflammatory, a pro-inflammatory microbiome populated primarily by proteobacteria, and Dr. O'Brien will talk a little bit about one of those particular cases. And this pro-inflammatory microbiome actually then leads to these autoimmune conditions that can lead to tissue damage And for example, in the case of cystic fibrosis, ultimately leads to the lung dysfunction and potentially to death in those cases. So the question now looking forward is, can we develop therapies that are based on the microbiome? Can we use our knowledge of the microbiome to use probiotic microbiome? So there have been now some recent publications that talk about transplantation of microbiome in inflammatory bowel diseases, for example, where they take the fecal microbiome of a subject and transplant it into another subject. And you can do it much more cheaply than using doctors, but we don't have to go there. And they've actually found that those transplants are beneficial for health, and they prevent further inflammatory conditions. And so now we're really looking forward to how can we use this knowledge to prevent and promote health. So, I'll take questions afterwards.
So thanks, Liliana, for starting the discussion off. Just to give folks here a little bit of a sense of the complexity of some of these microbiomes, I think I read that the gut itself has 500 or at least hundreds of identified, right?
In some cases, 750 to 800 different species.
Right. So the ecological complexity that we're talking about here, both in the number of species and in the numbers of cells themselves, is really absolutely astounding. So now we're going to drill in to Escherichia coli, which is one of, I guess, Alison O'Brien. You love this organism, right?
I love it.
You love it. So you're about to hear from someone who loves Escherichia. Ooh, okay, we're all eating it. So Alison, come on up.
Hi, thanks for having me. Can you all hear me? All right, I do a lot of teaching, but not to undergraduates, mostly to medical students and to graduate students. And I always tell medical students whose eyes are glazing over before I walk on that microbes rule. And you already got that impression. I hope you know and appreciate that your friend, the microbe, is dominant in your life. I'm going to talk about one particular kind of microorganism called Escherichia coli. Named after a pediatrician, the genus, the first word, Escherichia, species coli for colon. So when somebody says Staphylococcus aureus or Staph aureus, Staphylococcus is the genus, aureus is the species. E. coli, the pamphlet up here that we in the, from the American Academy of Microbiology helped write, really focuses on the good side of E. coli and the bad and the deadly. I'm going to spend most of my time on the bad and the deadly, but I don't want to shortchange E. coli. So E. coli is one of the microbes in your GI tract, in all of our GI tracts. It is a small fraction of our microbiome, but it's culturable. It can be grown on a petri dish. Clinical laboratories can grow it pretty quickly, and that makes it easier to study. For your information, the agent that causes tuberculosis also can be grown in the lab, but it takes weeks to culture. E. coli is culturable overnight. So the good side is it's easy to study, it's easy to grow. It has been the workhorse for molecular biology in many ways. We put things called plasmids, circular pieces of DNA, into which we paste genes and get them to express their transcribe, what's called transcribe and translate their proteins for the good of humanity to help make products, pharmaceutical products. They can be made in the vessel of E. coli in the lab. The metabolic processes that E. coli undergoes to replicate are very similar to the ones in our own body. So what happens in the elephant, so to speak, happens in E. coli, but it's sure easier to study an E. coli organism than an elephant, and I do not want elephant poop in my lab, so it makes it easier. Now, E. coli, a small fraction of what's in our gut, normally does not bother us. We have what we call normal flora E. coli. Sits with us, part of our microbiome, not doing anything terrible, maybe helping us, in fact, by making some vitamins, getting the oxygen concentration just right for other microorganisms in the environment. So E. coli can grow and breathe in the air on a petri dish, or you can stick it in broth and put the liquid all the way to the top and put a plug and make it without air, and it'll grow there too. It's what's called a facultative organism. But it will use up the air in an environment first. That's what it prefers to do. And it will make the environment without air for our GI tract microorganisms, many of whom are strict anaerobe, what we call strict anaerobes. They don't want to be around the air. They're killed in the air. So E. coli helps them out by setting the environment right. So E. coli is good to help us clone and express organisms, to understand metabolic processes. It helps other organisms in the GI tract, our normal flora E. coli. But E. coli, like the analogy in this little pamphlet, can be like a basic car. There's your basic version in your GI tract. And it normally doesn't cause any problems, but in a healthy person, particularly for the women in this room, if it escapes that normal flora, mostly normal flora E. coli, escapes from the gut and gets into the bladder, because of the anatomy of a female with a shorter urethra than a man, that is, that tube from the outside to the bladder is short, it's easy enough to get E. coli into a bladder. Women talk about bladder infections, and that's almost always caused by E. coli in our GI tract. Very common problem for women. 50% of women have had a urinary tract infection in their life, and sometimes more than one. Honeymoon cystitis. So that's E. coli in its normal setting, and occasionally when it escapes, or if something happens to us, we have somebody stick a rod in our GI tract, or we get a perforation, whatever's in the GI tract can spill out of our gut and cause major problems because it gets where it's not supposed to be. Normally it's sitting there in a healthy person, but if it gets out of where it's supposed to be, it can cause problems. But then there are E. coli that have different flavors. They pick up genes from other microorganisms that encode products that can be toxic to us. So I study a particular kind of E. coli that has picked up genes that encode a potent poison called Shiga toxin. How did the microorganism I'm studying pick up those genes? The genes are present on a bacterial virus called a phage, and there was a picture when you came in of one such phage or bacterial virus on the screen. So we have viruses and bacteria have viruses, plants have viruses. So the virus infected an E. coli at some point and stayed put in the E. coli in a quiet state, and it stays put that way until it's stressed, and then it can replicate, and it can replicate and make many copies of its genes and its gene product. In the case of the E. coli I study, it's this poison Shiga toxin. So you're thinking, who cares about this bug? Well, that's the Hamburger bug, you know, the bug you've heard about in Hamburger when Hamburger gets recalled, the bug you heard about in a big spinach outbreak, and maybe the bug you heard about in the Germany outbreak, foodborne outbreak last summer. You all heard about that one that involved thousands of people in Europe. And they, remember the cucumbers that got tossed because they thought they were the ones that were infected, and it wasn't the cucumbers. It turned out to be the sprouts. And then the Germans and the Spanish, they fought about who was responsible. And it turned out it was really Egyptian sprouts that were responsible for this whole thing. Okay, well, so that organism, that really inflamed people, got them interested, because so many people got sick. About 800 people got a disease called hemolytic uremic syndrome that put them in the hospital and put them in danger of losing kidney function. And it also killed about 80 people. And these were otherwise healthy people who had the misfortune of having sprouts put on their sandwiches, and they didn't even remember eating these sprouts. The sprouts had become, the seeds had become contaminated with an E. coli, type of E. coli, that had been infected with that kind of phage or virus I told you about that made a particular kind of this poison, type of this poison called Shiga toxin type 2. What was unusual about that outbreak is the backbone E. coli that got infected with the phage was not the typical backbone E. coli that causes the hamburger outbreaks or the spinach outbreaks or the cookie dough outbreak, actually, in the US. And so it was confusing. There was a concern that it could have been man-made. There's always this fear of man-made microbes. In general, we microbiologists think nature does a better job. But in this case, it was nature. It was a human isolate of E. coli, not a cattle isolate, which lots of the hamburger isolates are, that had gotten infected with the virus from another E. coli strain, and it had made an E. coli strain that was caused mild diarrhea into something that caused significant illness because it made a lot of copies of this toxin. So that was nature doing it, and it was human, we think, human fecal contamination of the sprouts, not cattle or manure contamination that led to this outcome. And sprouts are great for foodborne illness because they multiply and the bug loves the environment and replicates well in sprouts as they germinate. So this bug, the Hamburger bug, is called an adulterant in hamburger, and it can be responsible for recall. There are other variants of bad E. coli, one that causes Montezuma's revenge. Those of you that have been unlucky enough to get diarrheal illness on traveling, that's usually because an E. coli got infected with a circular piece of DNA called a plasmid that makes a toxin that leads to watery diarrhea. That make you feel bad. It usually doesn't lead to as bad an outcome as the outcome in Germany. So what I want you to appreciate is we have this big microbiome with lots of different pieces. We have studied some of the components better because we can grow them in the lab quickly, and we have used them as tools, but they still can cause disease under the right circumstances. And in the case of foodborne illness, it's usually the otherwise healthy person that gets hit. Sometimes the people that get hit the worst are those that also have disabilities. And with that, I think we can open the floor to questions.
So I just want to thank Alison and Liliana for hopefully getting us all interested in asking questions and has catalyzed some thoughts. What I ask is that if you have a question or some input, raise your hand. I will come with the microphone. Please do, if you're so inclined, identify yourself because we all find each other interesting. That is not an obligation, but if you can, that would be terrific. And I'll come around with this to help you ask those questions. I see one right here.
My name is Mike Forrest. I'm a science writer living in town. So I'd like to ask two questions for both or either of you. The first is about antibiotics. Thank you. The first question is about antibiotics. What is the long-term effect of taking antibiotics, which I understand clear-cut the microbiome in the intestine? And second, is it beneficial to take probiotics? So those are my two questions, antibiotics and probiotics. Thank you.
Okay, from a microbiome point of view, we can actually see the effects of antibiotics as far out as 3 weeks. I know some colleagues think that the effect on the microbiome, so let me backtrack a little bit. When we look at the diversity of the microbiome after exposure to antibiotics, it certainly diminishes. Okay, so different antibiotics will target different kinds of microorganisms, and each one of those antibiotics then affects the microbiome according to what microorganisms were killed off. So as I said, we can then study the progression of basically the recovery of the microbiome, and as I said, many cases, 3 weeks. Some people think it takes as long as 6 months to get back to a normal, stable microbiome that you would have started before the antibiotic. There are, of course, very significant effects. Go ahead on them.
I was going to say, when you need to take one, sometimes you just have to take it. And I think as was just stated, different antibiotics have different effects. Some are called what we call broad spectrum, and they will wipe out a larger number of microorganisms. One of the best antibiotics we have is penicillin. Is just not very useful anymore. Only certain organisms are still sensitive to it. So I would say, and physicians are being taught not to over-treat with antibiotics, not to treat every ear infection or every sinus infection with an antibiotic. Even though microorganisms are involved in the evolution of those diseases, they clear themselves often, and only under certain circumstances to use antibiotics. It's the overuse of antibiotics that has caused resistance. But our microbiome is affected by the use of antibiotics and takes a while to recover, as was stated.
So before I hand this off to the next questioner, and I do hope to see some more hands there, that question made me wonder about some products that we have in our houses and in our lives. This morning, in fact, I saw a bottle of Listerine in the bathroom, and it says, kills germs that cause bad breath, which sounds like a pretty good thing. But on the other hand, it makes me wonder, given what we've been learning here about maybe the changing attitude we have to our own microbiomes, whether that is a good idea. I mean, have there been generations using Listerine that have actually been doing harm to themselves? And in addition, now we have other kinds of products out there, all these antimicrobials and all these kinds of things where we're removing some portion of the microbiome in our environment. Can you just, given what you've been saying and learning, how should we think about these products?
Go ahead. Well, there's a hypothesis called the Hygiene Hypothesis that basically alludes to this overuse of antibacterials, antimicrobials, making our household so sterile to the point where our immune system is no longer being trained and can no longer really learn how to deal with microorganisms in a healthy way. And so the Hygiene Hypothesis states that in this sterile environment, as a child is growing and the immune system is being trained, it actually loses that exposure and it doesn't learn how to self-regulate. And so as a consequence of that sterility, now we have this increase in autoimmune diseases and hypersensitive responses, allergies, et cetera, that are a consequence of not having initial training. There are some data that support this, and certainly there have been some studies that are attempting to elucidate this, but we have a worldwide phenomenon of people using antibacterials and antibacterial soap, et cetera, so it's hard to do a controlled experiment in that case.
But I'm wondering if go ahead, Alison, before I
just wanted to add one thing. The best way to prevent a microbial infection from moving from one person to another in a hospital or from a cow to me or my child is to wash my hands. I don't have to use harsh alcohol. I don't have to use iodine, just soap and water. And in a laboratory that works with microbes, that's the first thing we learn. You got to wash your hands. If you're not, you can wear gloves, but you've got to wash your hands because that's the way you prevent infection. But that's like your old ivory bar of soap like people have been using forever, not this quite stringent kind of antimicrobials that are on the market now. And that doesn't last anyway. It doesn't last. The flora comes back.
Right, and just before I hand this over to a person with a question over here, Liliana, I think you were pointing out, talking about some kind of autoimmune conditions like Crohn's disease and such. Is there epidemiological data out there that shows that countries with certain kinds of hygiene and cleanliness characteristics, you might say, are not as clean as the ones in other countries that would be characterized as clean? Do you see different incidences of those kinds of diseases?
Yeah, so part of the reason the hypothesis has been postulated is because we see that in countries that are developing countries, basically, the exposure of children to to that sterility is not as prevalent as it is in countries such as the U.S. and other developed countries. And so the level of asthma and allergies in those countries is less than it is, the prevalence is less than you see in developed countries with higher levels of hygiene or sterility, I guess. But
That is true, but in developing countries, the second largest cause of death in children under 5 is diarrheal diseases, and that is in part mediated by microbes. So it's always a yin and yang. I think we both agree it's a yin and yang. There are the bad guys out there that you want to keep the innocents from, and there's the over attempt at cleanliness that probably, I think, does result in maybe enhanced levels of asthma and other effects that our immune system, because our immune system hasn't seen some of these microbes. So it's the balance. We've gotten rid of some of the problems of kids dying of diarrhea in the U.S. Instead, we have them having asthma. So yin yang.
Okay, and I should point out to those who are watching via the live stream, and I hope there are thousands of you out there, that you can ask questions by way of Twitter with the hashtag followed by Duxwa, DCSWA, and I will try to monitor any questions that come in the next 10 or 15 minutes. So do give that a try. Okay, next question.
Hello, I'm Harvey Lyford. I'm a freelance science writer, but not a medical writer. My question is, could one of you or both of you say something about C. difficile, in particular why it is so difficile and resistant to all apparently but 1 or 2 antibiotics and not very well to them either?
You want me to do that one?
Please.
Okay. That lucky for you, I guess, or not so lucky. That's one of the areas I talk to medical students about. So C. difficile has one particular attribute that makes it a really hardy survivor in the environment in the hospital or in a nursing home. So C stands for Clostridium. Difficile is difficult. Clostridium difficile is one of those anaerobic organisms. It lives in some guts of people in this room probably happily, and other people it can cause problems. In the environment, which is often the source of infection in a nursing home or in the hospital, the organisms exist as spores. These hardy, round, refractile bodies that can't be easily killed by handheld disinfectants in a hospital. Spores were distributed in the anthrax incident. Spores are made by Clostridium difficile. So that makes them different than many microorganisms. Secondly, if they end up in our GI tract and we treat with a broad spectrum antibiotic, they have the genes to make products that allow them to resist those antimicrobial agents, and they have a leg up and outgrow the normal flora. And then they make toxins, these poisons, that affect cells that affect the GI function. Interestingly, the fecal therapy that was mentioned earlier is a therapy that has been used in people that have repeated infections with C. difficile as a way to repopulate the GI tract with normal flora. So the answer to your question is the hardiness of the microbe, the ease with which the spores are spread, and the advantage the organism has in the gut when typical broad-spectrum antibiotics are used. And there's a highly resistant strain of the organism. Strain means a very particular type that is out there that has caused big outbreaks in hospitals. And it's more resistant than some other strains to a particular antimicrobial agent that's used a lot in hospitals, fluoroquines.
Victor Goldberg, and first a comment and then a question to Liliana. The comment is that, interestingly enough, today in the program Fresh Air, the same subject was discussed. And the advantage of that is that it can be downloaded at any time and you can listen the same things again. Now, the question to Liliana, are there things like color of skin or cultural background, like diet and where people are dressed, that affect that microbial population in the different parts of the body differently for different populations?
So we know definitely there are many cultural and specifically diet habits that change the microbiome in a healthy human gut, for example. So we observe differences in the microbiomes between people who have high fiber and high vegetable fruit diets versus a typical Western diet that's high in fat and high in red meat. And so those two microbiomes are basically, if you look at the data, you can tell right away which kind of person, which kind of diet that person has, just because of the types of microorganisms that you find in each kind. I would venture to say that tight clothing probably also changes the microbiome in your skin, but I don't think anybody has done that study yet.
I just did copy a paper out of the Proceedings of the National Academy of Sciences with the title Anthropology of the Microbiome, which was really looking very carefully at just these sorts of ideas and how there are regional differences and nutritional differences that affect the character of the microbiome.
I should mention that in addition to cultural and dietary, et cetera, behavioral changes, probably the most significant impact on your microbiome is actually your own genetics. And so you are a bad host, as Ivan was alluding to earlier, or you may be a good host. And so some of the differences that I mentioned earlier, I think are more likely attributed to your genetic differences between people than they are to particular behavioral conditions.
My name is Jamie Rosen. I work for a nonprofit organization that's developing new vaccines against tuberculosis called ERIS. And I understand from my colleagues just what you guys are saying, that well, related to what you're saying, that there may be geographic differences in how people's immune systems would respond to the vaccines that we're developing. And so I guess I'm wondering if you know anything about how that would work, and what other implications there may be if you have, a different microbiome.
Well, we're actually asking that very same question, except for Streptococcus pneumoniae, and we're looking at what are the differences in the response to the vaccine and whether the microbiome has an effect in that response. We are just now collecting the samples, and so I can't answer the question yet. But I think it doesn't necessarily, well, I don't know how much of it is affected by the geography, but rather, and as Alison was alluding to earlier, in different parts of the world, you have different strains, different types of the organisms. And so the strain that was available in Germany wasn't in the United States, it's just geographically it wasn't here. And so you see that the response to a vaccine might be different not only because of a host environment, but also because of the type of strains that circulate in that particular geographical location. So.
As well as something you already know, the HIV status of the patient would make a big difference, particularly for that vaccine. Yeah.
Certainly tuberculosis.
Yeah.
Hello, my name is Jim Johnson, and I have a question about this area called quorum sensing that I understand is being thought of as a mechanism for developing new strains of vaccines to prevent infections by various kinds of bacteria. Can you discuss in some detail exactly how quorum sensing and this whole idea of bacteria communicating among themselves work?
Certainly. You want to do it? Whichever. So quorum sensing was, I guess, initially discovered by Bonnie Bassler, maybe 25 years ago now? Somewhere. But basically what was found is that bacteria are able to produce and secrete molecules that can be sensed and detected by themselves and by other bacteria like themselves. And so upon sensing quorum sensing, what happens is if there are enough of those molecules floating around in the environment, then that triggers a response. If there isn't enough of that molecule in the environment, then that doesn't trigger the response. And so that's where the quorum comes from. There have to be enough bacterial cells to actually produce enough signal to initiate a cascade of reactions. In many of the virulent pathogenic bacteria, what you see is that quorum sensing regulates the virulence genes. And so we like to think of it in a very naive military point of view, that they're not going to go attack unless there's enough troops behind them to actually do any damage, right? So if there's not enough of me, I'm just going to hide out and wait till enough of us are around to actually be able to colonize. So there are at least 3 and potentially 4 different mechanisms for communication between bacteria. Some organisms have as many as 3 of those different mechanisms to communicate within each other. And we're now starting to understand that there is an interspecies communication. So E. coli doesn't produce its own quorum sensing signals, but it actually can detect the quorum sensing signals produced by other organisms in the gut, and so it can say, wait, there's too many of you, or wait, there's not enough of us, or whatever, signals like that. We also have learned that there's an interkingdom communication happening. So we're studying the response of pathogenic fungi to bacterial quorum sensing signals as a reactionary to, so what we think is happening is the fungi are producing antibacterial compounds when they sense too many of the bacteria around. Similarly, our human epithelium appears to be able to sense those signals as well. And I don't know that it's very clear yet how our epithelium is responding to those, but it can sense it as well.
Any progress in terms of building vaccines?
I don't think so.
I don't think so either, but I don't know. Not of enteric organisms, if that is the actually approach that's being successfully used, I'm not sure.
I don't think so.
All right, in a first ever, I'm going to go to a question that has arrived online through Twitter, by way of this live streaming. So this is exciting. This is coming from, I believe, just based on the name of the Twitter handle here, which is Amateur Micro, that is someone who is an amateur scientist who probably works in a basement. And he's interested in microbiology and microscopy, he or she. And the question is, what resources do you recommend for learning to do it safely so as not to kill the entire neighborhood? I'm adding that, but...
Well, there is a lot of concern about basement microbiologists. People, really there are concerns. I would say stick with E. coli that could be used as a cloning vector. That is very unlikely to ever cause disease in anybody. Yes, ma'am.
We have a resource, microworld.org. The American Society for Microbiology maintains a website, microworld.org, and on there we have lots of activities that students, children can do, and they are all microbiologist approved, and they're all safe. So please use that as a resource to learn some more about microbiology.
And don't lick the plate.
Yeah. And wash your hands.
Hi, I'm David Shapiro, and the question I'm going to ask comes out of the fact that a while back my sweetheart got awfully sick because she ate some chicken that was left out too long by the restaurant, and it's not that she got infected, but that she was subjected to the toxins that had been generated by the chicken. So my question is, pink slime. These products are sterilized, as it were, by exposure to ammonia or an ammonium product. Does this mean that they're safe, or is there an issue that this is not really addressing properly? I don't care that the idea may be disgusting, that the name is pejorative.
I can't answer because I don't know about how sterile pink slime is. I do know that Hamburger is absolutely not sterile. Chicken is absolutely not sterile. And that most of the problems, I'm not sure about how it happened with your sweetheart, that they could have had raw chicken next to her lettuce and the juice could have contaminated her. The lettuce, it was probably Campylobacter. That is the name of an organism that often causes infections associated with poultry. It could have been another bacterium called Salmonella. Neither of which produce toxins or poisons, but they both can cause pretty serious illness. The answer about the pink slime, I can't say, but for Hamburger, when I used to eat Hamburger, and I will tell you the reason is not what you think about why I don't eat Hamburger anymore, but even though it may look cooked, it isn't, and there's still organisms in it. You have to have the right temperature. That was a lesson learned in the Jack in the Box outbreak in 1993, that it's not just the color, it's the temperature of the meat that kills the microorganisms. But the reason I don't eat Hamburger, or at least not very often, is because I was on the USDA panel for microbial food safety, and they made us look at how animals were slaughtered. So that stopped that particular food for me, not what I was working with.
Hi, my name is Aaron. I had a question for Alison. You were
Aaron.
talking about the way that microbes change, and you mentioned in menstruation for women that there was a change, and so I was just curious, what would this change look like, or can you talk a little bit more about how you see the change, or what is this change that?
Okay, and that was not me that mentioned that change, but I'll make one little point, and I'm going to turn the microphone over to her. Women are more susceptible to gonococcal, Neisseria gonorrhea, that causes gonorrhea. They're more susceptible at different times in their menstrual cycle. That may be hormonal, or it may be differences in the flora.
So actually the female body as a whole changes pretty radically during the whole menstrual cycle. You see differences in the kinds of organisms that survive on all of the skin. You see very radical differences in the pubic area, the pubic skin, and also in the vaginal tract. So normally you would have during the non-menstrual part of the cycle, a woman, for example, has a very robust lactobacillus community in her vagina. And during the menstrual cycle, that actually shifts, the pH shifts, the lactobacillus doesn't survive as well, and the microbiome is actually different in those cases. So there's some very nice literature that discusses the shifts certainly in the vaginal microbiome during menstruation and during the whole cycle. Out of the University of Maryland, Baltimore.
Hi, my name is Linda Voss. I write about science and technology for NASA.
Oh, there she is. I see her.
You were commenting about the discovery that microorganisms cause disease. I'm assuming that people are looking at cancer as a disease, and I'm wondering if there has been any correlation found between the microbiome and the promotion or inhibition of cancer.
I'm glad to answer that one. Well, let's start with the one carcinogen. Microbial carcinogen is called Helicobacter pylori. It is the organism that's in our stomach, in many of us, not all of us in this room, and it's been linked to gastric cancer. It's the only microbe so far that's been called a carcinogen. We do think that there are changes in the microbiome may be promoting inflammation, which may promote cancer.
Yeah, absolutely. I think that right now our understanding where we are right now is that aside from Helicobacter and of course many different viruses that may also be involved in producing cancer themselves, it is the effect on this chronic inflammation that leads to these very wide range of diseases, including, as I said, liver diseases and potentially cancer. So.
I have a question. Crohn's disease is a very odd disease in many ways, and it's different in many ways from other bowel diseases.
Where is she?
Is it caused by a bacterium, a virus, a bacterium infected with a virus? The causes that have been associated with it have, or its appearance, or heredity, ethnicity, climate, exposure to light, all kinds of things. But its behavior is so odd. How much knowledge is there about exactly what does cause it? And is it possible to even know that?
We're talking about Crohn's. Okay, I didn't hear it. I don't have an answer.
Well, I'm not an expert in Crohn's. I should start by saying that. But what I have read is that what happens in Crohn's is, again, a constant inflammation. And one of the reactions of your epithelium to this chronic inflammation is normally the epithelium is very tight. The cells are held together by a very, very tight cement. And so what happens during inflammation is that they loosen up just a little bit, because one of the objects of inflammation is to bring in immune cells that can make their way through that cement. Right? So they want to loosen up so those immune cells can come out and actually grab all the microbes that are floating around and making you sick. So, because that's what they think is happening. That's what your cells think is happening. So as those tight junctions are loosened, that actually makes it more prone, makes an epithelium more prone to diffusion of other molecules that are floating around that shouldn't be coming into the bloodstream. And that interplay between what's coming out, what's coming in, actually leads and conduces to tissue damage, local tissue damage, that then results in the effects that you see in the Crohn's, in the symptoms in the Crohn's disease. So, there's not one particular organism. We don't know that there's a specific etiology for that, except for potentially the inflammation that's leading to that loss of the tight junction.
Does it become a problem for people that microbes have acquired resistance to antibiotics?
I'm sorry, I can't hear you. Could you stand? Or could you, here we go.
Microbes have developed resistance to antibiotics. And in some countries, people use bacteriophages to treat microbe infections. Is it more difficult for microbes to
I'm sorry, I'm getting some of the words. No, I'm not getting all of the words.
Yeah. Yes.
Okay.
I think she's asking a question about phage therapy, which is quite interesting.
Yes, I'm happy to try to answer
preceded
that.
Antibiotics. It's a very interesting story.
Yeah.
Is that your question? Okay, so maybe you can tell the
Okay,
group
I'm happy
about that.
to give it a shot. So phage therapy was used in this country in the 1920s.
Actually, can you
First off,
once again
phage is
say
a
what a phage is?
bacterial virus. A phage is another name for a bacterial virus. So these viruses, like viruses that affect us, sometimes they don't affect another animal. Some viruses affect only staphylococci. Some viruses only affect certain types of E. coli, but not others. They're pretty specific. And they attack from the outside and often inject their nucleic acid in the organism, which sometimes then becomes part of the microorganism's DNA and sometimes causes the organism to pop. What we're talking about is hoping to inject and cause to pop and kill with phage therapy. That's what we're like, injecting a needle and hoping to get the bug to die. So that phage therapy was used in the 1920s there were outbreaks of staphylococcus, which you all know is a pretty common organism. Most people have it on their skin, but it was causing major infections in nurseries in New York. And so the thought was that if they could use this was pre-antibiotic, specific tools that would target staph, like specific viruses for staph, they might be able to get rid of the microorganisms on the umbilicus of the babies and not let the bacteria, the staph, get into the bloodstream and harm the infants. It has been thought that maybe phage therapy could be used to cure cattle of the Hamburger bug, which we call 0157H7. That's just a label because of the outside of the bug. Phage therapy has been used in a very elegant way by a man named Vince Fischetti, who is a member of the American Society for Microbiology, a prominent member who's at the Rockefeller. He studied the organism that causes strep throat for a number of years. We call it strep, or group A streptococcus. He has found, he and his collaborators in his laboratory have found, if you take the phage apart and you look at the gene products that cause that popping, and you work with that product alone, you lose some of the problems of phage therapy. And the problems of phage therapy are that bacteria become very resistant very quickly to those phage because they change their surface. They're very wily. There are a lot of them, and they're very wily. They change their surface, and by changing their surface, they don't allow the phage to attach. But if he gets rid of that part of the cycle and just uses what he calls the lysins, he has very specific effects on Bacillus anthracis, group A streptococcus. So he has a new venture company that he has developed using this component, this particular gene product for certain particular phages that bacteria do not develop antibiotic resistance to. So I think that the time of phage therapy may have come around, and maybe we're at a different level than we were using whole viral phages. Instead, we're using gene products from them. Okay?
Okay, and I just want to point out, too, that if anybody is so inclined to go look on the web at what phages look like, they are magnificent structures. Their heads are these amazing geometric forms that house the genetic material. The T4 phage, which I've looked at, looks a little bit like, for those of us who are old enough, the Moonlander. The Moonlander. And then it has a kind of injection system. It's almost like a molecular syringe system that when the phage alights onto a bacterium, there's a structural change. It happens, a little twist occurs in that syringe that then plunges the genetic material into the bacterium. It is absolutely just amazing from sort of the perspective of nanoscopic machines.
What I think from microbiology point of view, when we started sequencing microorganisms, we were astounded to find out what percentage of a microorganism they were bits of phages from other organisms, or whole phages that had integrated, become part of the genome of that organism. For the Hamburger bug 0157H7, there were many, many phage components, and they bring DNA into the microorganism and change it in certain ways. So phages, these fantastic looking apparati really do change microorganisms in terms of virulence traits, ability to colonize a surface differently than it could before it saw that phage that brought in DNA.
Hi, I was also curious about probiotics. That was a question before that didn't get answered. How good they are for reestablishing healthy flora. And I have a second short question. Do you think it could be, microbiome could be used for fingerprinting in the future, since there are these rare specific bacteria in our bodies?
So I think as far as the probiotic, the scientific jury is still out. So I think we're really just now trying to collect the data that supports some of the anecdotal observations that people have made. So I have my own anecdotal observations about not using fluoride toothpaste and seeing a drastic change in my nasopharyngeal microbiome. I don't get sinus infections anymore, for example. But that's just my own observation. I actually haven't done any scientific testing, gone back to using fluoride, see if that makes a difference. I mean, I have no idea what actually caused the effect. So as I alluded to earlier, I think just now we're really starting to do a systematic review and a systematic scientific testing of whether probiotics are truly giving that sort of beneficiary effect. The second question was fingerprinting. Yes, that is a goal. I guess one of the things that I tried to convey was that microbiomes are not static. They're variable. They change even within one individual. So I read a paper that did try to do a bacterial fingerprinting instead of an actual digit fingerprinting. But I don't think that it's possible to have any sort of long-term fingerprinting because that just changes too much.
We have a repeat questioner, Michael.
Yeah, I wanted to follow up on that question about probiotics. So if you take them, aren't you changing your internal microbiome in some significant way? And if so, what is the impact of doing that?
Can you change?
If you take probiotics, does that change your internal microbiome in any significant way? And if so, what is the impact of doing that?
So I am highly skeptical of taking the pills that you buy at, in the pharmacies that have 10 to the 12 different bacteria, and that those would actually colonize your gut. If they do, it would be a very minor population of those. You don't know how many of those are living. So as I said, I don't think there's any scientific data to support that yet. I do think that in the cases of antibiotic, when you are prescribed antibiotic, if you repopulate your natural microbiome with probiotic foods such as yogurt, other fermented products, it can actually help alleviate some of the discomforts associated. Sometimes you get diarrhea or other things when you're taking antibiotics. So I think in those cases when you are able to recolonize quickly upon the use of antibiotics, they can be beneficial.
Hi, my name is Dave Oberbillig, and the question I have is about this system of microbes in human cells. And as I understand it, bacteria are really good scavengers of genes in their environment, or they can be. So have bacterial cells over time taken on any human genes and or vice versa? And does that have any implications for the coevolution of us with our flora and or health or disease?
I think viruses are better and have done a better job at picking up human genes. And I think the reason for that is they're absolutely dependent on the human, for human cell, animal viruses to replicate. Bacteria, I would say, instead of taking up human genes per se, I was hunting in my head for what human genes have been taken up by bacteria. They hijack human gene products to do what they want to do. We've learned, we microbiologists came into the cell biology field very late, really, when we realized how many of the products injected into human cells from bacteria. Some of these are called type 3 secreted products. They get injected and they alter human cells and allow for a more comfortable environment for the microbe. We believe, there was a question about TB and TB vaccines a while ago. We believe Mycobacterium tuberculosis. That's a name, the genus and species of the organism that causes tuberculosis, really plays with the cell biology of the host cell so that it escapes being killed within a host cell. So I'd say bacteria are better at altering, adapting, or using, and viruses have picked up human cells or human-like cells for their purpose.
I think in addition to what Alison is saying, bacteria and other microorganisms have learned how to mimic the host very well. So they use proteins but they're bacterial genes that have been modified to mimic the host's the host proteins. Okay, so they'll mimic some of the cell surface so that the immune system says, oh, this is not infected cell, keep on going. So they'll do something like that. But talking about exchange between bacterial and human or prokaryotic and eukaryotic, there is, of course, a very nice study, a nice case between chloroplasts and mitochondria that were once free-living prokaryotic organisms that have since transferred almost all of their gene content to the nuclear genome of their host and have then become obligate organelles in those hosts. So that's the only story we know of. We think that there might be some molecular evidence of further organellization of some organisms, but we don't have the whole story yet.
Hello. Given that bacteria are swapping genes with each other all the time, how do you define a species of bacteria, and how do you classify? I mean, how do you know what to count, and what do you count when you're looking through the microbiome? Do you just extract and sequence DNA, and then is there a consensus about how to classify that? Or
It sounds like there are two different issues. One is the old-fashioned way of classifying them, like a stain called a Gram stain. And many of the organisms actually fell out correctly. Some didn't. But she's going to tell you how they do it with 16S ribosomal RNA, or other ways to classify and put bacteria into bags. But from a medical perspective, E. coli to us is still E. coli. Shigella is highly related to E. coli. Yeah, we call it still. People will argue and say it should be called E. coli, but it causes a different disease in people. So they've kept the separate names, but truly it really is E. coli. But the old-fashioned way of separating was based on what we saw and what happened in the host. And humans were very centric toward humans. But I think that with this new world that's being examined, there's a different perspective, and you should give that perspective.
Oh gosh, that is, I don't know how many $1 million question. So what we're doing right now, okay, and this might change in 5 years, this might be static, yeah, I have no idea. But what we do for microbial classification, bacterial and archaeal both, is we use the ribosome gene. So the ribosome is a machine in every living cell that produces all the proteins. Okay? And the ribosome function is, of course, very conserved because the protein synthesis is the same across all organisms. So because the structure of the ribosome is conserved, what happens is the underlying genes that encode for that structure have also been highly conserved. But over the course of evolution and speciation, that those genes have been modified slightly. And so basically what we say is that a species has its own ribotype, meaning that it has its own sequence of those genes. Okay, and so that's the classification. You have to have a unique, basically, 16S sequence or ribosomal gene sequence to be classified as a species. So if you share the same sequence like E. coli and Shigella, then you're the same species, even though phenotypically they might be classified differently.
We only have a few minutes left, and I will go to the question over here, but before we do close, I just want to turn the question back toward, or a question toward you as scientists. Of all the things you could have studied, you both became microbiologists. I'm just wondering if there was some teacher, some life experience, something that directed you into the particular area of study that you now have become experts in.
Well, I'll speak first, as I'm the older of the two of us. I would say there were three things. One was my love of reading, and I had a mother who was a librarian who used to bring me home all these books. And I loved something called the Microbe Hunters, which you probably never heard of, right? I read Aerosmith, and I loved that scientist in Aerosmith following. He was a phage biologist, actually. And the third was a teacher. I was a biology major, and I was thinking about majoring in, it was between math and biology, and I took this two-unit course in the vet school in pathogenic bacteriology, and that was it. That was it. I knew this is what I love to do. So it was a combination, but it was that class, that one class, that made me really, really like it.
That's actually very interesting. So if you have kids in college and you want them to decide to be microbiologists, take them to a bacterial pathogenesis class, because it's the best class they'll ever take. And certainly that had a major impact. I had grand dreams of making, believe it or not, however many years ago, of making cellulolytic ethanol. I should have stuck with it. But in my pursuit of that, I studied biochemistry, and one of the courses that I took in biochemistry ended up being bacterial pathogenesis, sort of to fill up the credits that were needed in the biology. And it changed my life forever, so.
I think we have time for one more before we close, and I do ask your forgiveness for all this terrible feedback we've been experiencing.
Hello, I'm Hannah Waters. I'm a science writer. And my question kind of combines the species question with something that was mentioned briefly in the intro, which is the Craig Venter projects in the Sargasso Sea. So, as I understand correctly, those projects are, instead of trying to break down bacteria into individual species, it's just kind of scooping up a bucket of seawater, sequencing the genes, and just not caring about species and just saying, these are the different functions that are being performed. Is there any research going on trying to do that with the microbiome? And what can we learn from that? Is that going to kind of lead to any kind of therapeutics?
Yeah, certainly. So there's two approaches that are used in the microbiome projects. One is to just look at the class roster, basically using 16S sequencing. So we just get a list of hundreds and thousands of microorganisms in any particular body site. In addition to that, we're also doing whole genome sequencing of everything that's there, like you suggested, to try to characterize at least the most abundant genetic composition of that microbiome. So both things are used together. We know from 16S sequencing we will get some idea of the metabolic capacities that are there, and we also, of course, know that phage, plasmids, other mobile elements have an important role that is totally undiscoverable by 16S sequencing. And so both approaches are being used, and I just read a fantastic paper of a system biology approach to characterizing metabolic capacity of all the different sites and differentiating disease and non-disease using a whole genome approach. So definitely we're getting a lot more understanding of diseases that way.
All right, well, with that feedback, I think we need to close now. We're at the end of our time here. So a hand to both of our discussion leaders. Thank you, Liliana. Thank you, Alison. I do encourage everyone who wants to be kept apprised of these cafe events to sign up on my email list. Thank you all for joining this discussion, and come back on the April 25th. Again, another hand to our discussion leaders.
Thank you, audience.